Here is an overview of the urinary pathway and the ejaculation pathway. Now, the urine is produced in the kidneys. We have two kidneys, therefore we're going to have two ureters that are going to bring the urine to our bladder. Once the urine reaches our bladder, which is located over here, it is going to be taken out of the bladder and secreted out of our body through a tube called the urethra. Right, so this is considered the urethra. Now, depending on where the urethra is, the name is going to change. Right below the bladder, we have the prostate. The urethra that is found in the prostate is called prostatic urethra, which makes sense. When the urethra leaves the prostate and is in the membranous region, it is called the membranous urethra. When the urethra enters the penis, it is called the penile urethra. And from the penile urethra, it can leave the body. Now, this was the urinary pathway. Let's talk about the ejaculatory pathway. First, we need to talk about where sperm is made. Sperm is made in the seminiferous tubules. From the seminiferous tubules, it is going to reach the red testes. Over here. From the red testes, it is going to reach the head of the epididymis, and then it is going to travel to the tail of the epididymis, then ultimately is going to reach the vas deferens. If we go back to our previous image, we could see the head of the epididymis over here, then the tail of the epididymis. Then it is going to travel through the vas deferens, which is this tube over here. Now, you can clearly see that the vas deferens is going to empty the sperm into the seminal vesicle. There are two things that we need to know about the seminal vesicle. The first thing is that it is going to contain the enzyme sorbitol dehydrogenase. This is an enzyme that is very high yield to know for US Assembly Step 1. We will talk about this later. The second thing we need to know is that the seminal vesicle is going to secrete alkaline solutions to the sperm, alright? And the reason for this is to neutralize the acidity, the acidic environment that is found in the vagina, so that the sperm can live there and survive. Another reason is because this creates an ideal environment for the sperm to function in. Now that we have secreted the alkaline solution, the seminal vesicle is going to secrete its contents to the ejaculatory duct. From the ejaculatory duct, it is going to reach the prostatic urethra. We talked about how urine from the bladder is going to leave the body, and the first tube that it is going to enter is the prostatic urethra that is found in the prostate. So this is the meeting point of our semen and urine, right? So the urine and semen can all travel through the prostatic urethra, and then it is going to both travel to the membranous urethra, and then it is going to reach the penile urethra before leaving the body. Now, how do we determine whether we're going to release semen or urine? Well, if we were to release semen, before releasing semen, our bulbourethral glands play a role, as you can see over here. So before ejaculation, bulbourethral gland is going to release pre-ejaculatory fluid to remove urine. Bulbourethral gland is also called the Cowper's gland, and this pre-ejaculatory fluid that it releases is also called the Capper's fluid. The Capper's fluid is basically a alkaline solution that is going to neutralize the acidity of the urine. Why is our urine acidic? Because we have urea from protein catabolism in our urine. So it is going to neutralize that so that it doesn't kill our sperm cells. Now, let's talk about sorbitol dehydrogenase. We said that seminal vesicles contain sorbitol dehydrogenase. Why is this important? This is very important in fructose metabolism. This is the polyol pathway. You might have gone over this pathway when you were talking about diabetes. And essentially, when we have high levels of glucose in our body, instead of glucose going through normal glycolysis, it is going to be converted into sorbitol with the enzyme aldose reductase, which has a high Km. So aldose reductase only plays a role in the conversion of glucose to sorbitol when there's lots of glucose in the surrounding environment. So normally sorbitol can, or in diabetics, sorbitol can accumulate within cells that don't have sorbitol dehydrogenase, cells like, or organs like kidneys, lens and retina, Schwann cells, and it can cause damage to these regions through osmotic damage. Because sorbitol is a osmotic agent, it draws fluid, and that can cause osmotic damage. Now, the enzyme sorbitol dehydrogenase is found in our seminal vesicle, and this is very important because in the seminal vesicle, sorbitol can be converted into fructose, and fructose is the main energy source for our sperm cells. This is very high yield to know for USMLE Step 1. Fructose is the main energy source for our sperm cells. Fructose is used for the motility of our sperm cells. Therefore, it is going to be extremely important for our sperm cells to be normally functioning.